let's see the effects of various hormones on the lipid metabolism. So right now we are doing a comparative study of how different hormones act on the lipids and use this information to prepare a chart so that we can remember the effects better. Let's write down first the names of the hormones which we have completed. Starting with growth hormone, ADH, oxytocin, T3, T4, prolactin, calcitonin, aldosterone, cortisol, epinephrine, and norepinephrine. Out of these, now let's see which have effects on the lipid metabolism. GH has effects. T3, T4 have effects. Cortisol has effects. Epinephrine, norepinephrine has effects. So total 4 have effects. Now the remaining will not have effects. So let's see and count them. ADH, no effects. Oxytocin, no effects. Prolactin, no effects. Calcitonin, no effects. Aldosterone, no effects. So 5 have no effects. Now let us see what these 4 have effects on lipid metabolism and do we have a commonality between their actions. GH, T3, T4, cortisol and epinephrine all cause lipolysis. But let us see how different their functions are. In the end, they cause lipolysis but in different ways. So let's start with GH. GH causes lipolysis of adipose tissue, which increases the free fatty acids in the blood. It also promotes lipogenesis, where in the liver. GH is also called as a ketogenic hormone. Now, why it is called a ketogenic hormone? That's because there is increased oxidation of the FFA, that is the free fatty acids, which get converted into 3 things: ketone bodies, acetoacetic acid, and lastly beta hydroxy butyric acid. GH also increases the ability of the pancreas. I repeat, it also increases the ability of pancreas to react to or to respond to insulinogenic stimulation. That means agents which increase insulin release. So GH will increase the ability of the pancreas to respond to these agents. Which agents? The agents which increase the release of insulin from pancreas. The examples are arginine, which is an amino acids, glucose, and many more. Now let us see the effects of T3 and T4. T3 and T4 again cause lipolysis, but how? They promote cholesterol synthesis. They also promote lysis of cholesterol. That means first they form cholesterol and then they break cholesterol. Now, if the cholesterol is broken, some of it is used and the remaining is excreted. So they also promote biliary excretion of the cholesterol. Now, because of all this, there is increased release of two things: free fatty acids and glycerol. Now, this glycerol here is used for gluconeogenesis. I repeat, gluconeogenesis. Also, there is increased turnover by facilitating oxidation. There is increased LDH receptors in liver. So because of this, since there are more number of LDH receptors in the liver, there is increased cholesterol uptake. That means more amount of cholesterol is taken up by the liver. What is the net effect of all this? Net effect of T3, T4 on lipid metabolism? There is reduced plasma levels of cholesterol and lipids. Other lipids to be specific. Now let's see the effect of cortisol on the lipid metabolism. So again, cortisol increases the FFAs, that is the free fatty acids in the blood. How does it do this? It does it by increasing lipolysis. So more amount of lipids are broken down, and that is why free fatty acids are released into the blood. The free fatty acids which are released are now utilized for energy production. Okay. So fatty acid synthesis is also a part of function of cortisol. But the fatty acid synthesis does not take place everywhere. It is inhibited in liver. That means in liver there is no fatty acid synthesis. That means no fatty acids are formed in the liver, but it is not inhibited in the adipose tissue. So the effect of cortisol, fatty acids are formed not in the liver but in the adipose tissue. Because the fatty acids are formed, also ketone bodies are formed. Ketone bodies. So in general, let's summarize the effects of cortisol on lipid metabolism. The effects of GH and norepinephrine, which affects the lipolytic effects. That means GH and norepinephrine both have lipolytic effects, but these effects require a hormone, and that hormone is cortisol. This is called the permissive action. I repeat, GH and norepinephrine both have lipolytic effects, but their lipolytic effects require another hormone, and that hormone is cortisol. Also, during fasting, there is increased lipolysis, which increases FFA, and glycerol. Also, cortisol promotes ketogenesis in in diabetic patients. Now let's see the effects of epinephrine on lipid metabolism. So epinephrine, it also has an effect of lipolysis. That means it breaks down lipids. But how it does this? It does it via beta receptors. So when it does it via beta receptors, these beta receptors, they will mobilize of epinephrine on lipid metabolism. Epinephrine acts via the beta receptors. Now, when it is acting on the beta receptors, it will mobilize the free fatty acids. So when it is mobilizing the free fatty acids, it also does one more thing. It activates hormone sensitive lipases. Activates hormone sensitive lipases. It also promotes beta oxidation of free fatty acids in muscle and liver. In short, epinephrine is pro-diabetogenic and pro-ketogenic.